Today's lecture will be about pH and biomolecules. By the end of this lecture, you should be able to define pH scale, acid, base, buffer, carbohydrate, protein, lipid, and nucleic acid. Be able to give examples of acids and bases, and compare properties of different biomolecules, which would be the carbohydrates, proteins, lipids, and nucleic acids. First off, in class, we will be having a class activity where we talk about whatever was the most confusing from the last lecture. This is an online lecture and will not have that. So let's start off with pH. pH is basically how well a molecule can donate a hydrogen ion. If you remember, hydrogen is the first element. It has one proton, sometimes one neutron, and will have one electron. When you take that electron off, you make it a cation, or a positively charged ion. And we put this as H+. Sometimes I will refer to this as a proton, because if it is a proton and an electron that make up hydrogen, and you take away the electron, you just have a proton. So H+ or protons, these are hydrogen ions. And some molecules, when they dissolve in water, water being a solvent and them being a solute, they will dissociate into a hydrogen ion and the rest of the molecule. These are what we call acids. So acids are able to donate hydrogen ions. And this would be something like hydrochloric acid, which is HCl. You may remember NaCl, sodium chloride or table salt, dissociates into Na plus and Cl minus. Hydrochloric acid dissociates into hydrogen plus and Cl minus. That would be a hydrogen ion, and that would mean it's an acid. There are also things like bases, and these will be able to accept H+ ions, perhaps because when they dissociate, they make an OH, and an OH plus an H turns into H2O. So these are able to accept hydrogen ions. And this would be something like bleach or sodium hypochlorite. We're not going to get into the chemistry of that in this class, but it is a very basic substance. Stomach acid is another example of an acid, and Tums are a base. And when these are mixed, they neutralize one another, which means if you add one thing that can donate protons and something that can take them, then you put them together, they end up neutralizing and coming closer to a neutral pH, which would be 7. You will also see the term alkaline, meaning more basic. So that's just another term that we use. And this scale over here say concentrations of H+ in moles per liter. We're not going to get deep into molarity in this class, but this is a negative log scale, which means you see this 10 to the negative 1 for hydrochloric acid means basically 0.1. That's 10 to the negative 1, or 1/10. 10 to the negative 3 is 1/1000th, or 0.001, which is a smaller concentration than 0.1. And that means these concentrations of hydrogen ions get smaller and smaller as the pH increases. There are also things known as buffers, and buffers will prevent a change in pH. So you have a buffer in your blood. Carbon dioxide dissolves in blood and combines with H2O to become carbonic acid, H2CO3. Carbonic acid dissociates. It is a solute in the solvent of water that is in your blood, and it turns into H+ and HCO3-. It is an acid. Now that bicarbonate ion, HCO3-, can accept an H+, or it can donate one, depending on the pH of the blood. If the pH goes down, it starts accepting more, which brings the pH back up. If the pH goes too far up and becomes too basic, it can donate more and becomes more acidic. This resists any change in pH in your blood, which is good for you because you need your blood at a certain pH. It is a buffer. Buffers resist change in pH. So that should about finish our solvents, solutes stuff. Let's talk more about organic chemistry. Organic chemistry is the chemistry of carbon. And carbon is a building block for most biological molecules. So we will see carbon just about everywhere in biology. It's a very important thing. Each atom can form 4 bonds in a combination of single bonds, double bonds, and triple bonds. We will see mostly single bonds and double bonds to other molecules. You see carbon itself can take the form of diamonds or graphite depending on how its bonds are associated. We have carbohydrates. Carbohydrates are made of carbon, hydrogen, and oxygen. Usually a carbon backbone. Here we see the carbon backbone just being the little arrows that we have there. And you see OH and H connected to those. Those are your hydrogens and oxygens that are attached. So things like glucose and fructose are both sugars. Sugars are these single or sometimes double molecules. So a monosaccharide, glucose, or a monosaccharide, sucrose, can combine to make a disaccharide, which is two sugars together, and that would be sucrose. There are also oligosaccharide, which are few sugars, or polysaccharide, which are many sugars, and you can get from that starch. So if you keep on adding glucose molecules to each other, and you remove water every time you add two to each other, it's called a dehydration reaction, you end up getting starch. And starch is this long chain of sugar molecules, of glucose molecules. And you can always break some glucose off and use them for energy. We will talk about metabolism a little later. This energy is stored in the liver. Starch is an unbranched chain. We also have something called cellulose in plants, which has different bonds between the glucoses, and we are not able to digest cellulose. Lipids are hydrophobic. They are not soluble in water. We see here a triglyceride, which is made of 3 fats, each one of those little chains down there, and 1 glycerol molecule. That is a triglyceride. And we sometimes call these fatty acids. We'll show the structure of these a little later on. We refer to fats as both saturated fat and unsaturated fat. Saturated fats have all the hydrogens that they can. So what's in a lipid? Well, mostly carbon and hydrogen. No oxygen, no nitrogen. And if you remember from the last lecture, oxygen and nitrogen tend to make polar bonds. So these carbon and hydrogens, which do not make polar bonds with one another, make a nonpolar molecule that does not dissolve well in water. A saturated one is all the hydrogens that each carbon can handle. These are saturated with those hydrogens. An unsaturated fatty acid has double bonds between some carbons. It could hypothetically carry more hydrogens, so it is not saturated with the hydrogens. We will also look at things called phospholipids in cell membranes. These have, instead of a glycerol, they also have a phosphorus head on there, which is polar. That makes part of the molecule polar and part of the molecule nonpolar, and that's special for reasons we'll discuss later. Another type of lipid is a sterol. This is a lipid that is formed in the shape of a ring, mostly carbon and hydrogen bonds once again, and one example here is cholesterol. So we will see cholesterol in cell membranes, and we'll see it later on in some nutrition. Proteins are amino acids, shown here, linked together into long chains. There are 20 types of amino acids that humans use to make proteins. They all have an amino group, NH3, and an acid group, COOH, shown here as COO- because it has already donated its H. It is an acid. It gives an H plus away. These amino acids have different what we call R groups, and those are the things that are highlighted in this picture over here. Those R groups are on the side chain, and they are different for each one. So while all amino acids have carbon, hydrogen, oxygen, and nitrogen, some amino acids will have other things on the side, like sulfur in methionine or in cysteine. These are in different groups here. The amino acids with nonpolar R groups, uncharged R groups, uncharged but polar R groups, negatively charged R groups, and positively charged R groups. We have different types of these. And what happens is these will give different structures and allow different functions for the protein. So about protein structure, we have 4 different ways of looking at amino acids bonded together in a protein. The primary structure is just the sequence of amino acids, basically a list of amino acids that are together in a protein. A secondary structure is how these amino acids are put together. So we have an alpha helix as one potential structure for amino acids. We have a beta sheet with one potential structure for amino acids. Now, the tertiary structure is the structure of alpha helices and beta sheets in a larger single polypeptide chain. Polypeptide meaning many peptides or many amino acids put together. A quaternary structure is several different polypeptide chains put together. One example of this is the 4 polypeptide chains that make up hemoglobin in a quaternary structure. Proteins can do things in the body, and one thing they can do is be enzymes. Enzymes are proteins that do things. They are biological catalysts. They can alter the speed of reactions without themselves being used up. So these will aid in biological reactions like metabolism that we'll see later, or like making more DNA, which we'll see much later. They do a lot of things in the body, and we will see them quite often. These enzymes, remember, are proteins. Nucleic acids are used to store information or transmit information, as well as some role in energy. So nucleic acids are made of nucleotides, which is a 5-carbon sugar. Yep, that's right. They're made of part sugar, a nitrogen base, and phosphate groups. So the types of elements present in nucleic acids are C, carbon, H, hydrogen, O, oxygen, N, nitrogen, and P, phosphorus. Only molecule that has phosphorus out of all these biomolecules. There are 4 different types of nucleotides that make up DNA: adenine, thymine, cytosine, and guanine. There are 4 types that make up RNA: adenine, cytosine, uracil, and guanine. So you can see here, there are 2 different types of nucleic acids we will see most often in the human body. The first is DNA. DNA is in a double helix. This is a very stable structure that can stay around for a long time. You will see this in the nucleus. The nucleus has your genome. Your genome is made of DNA. This genetic code is basically the sequence of nucleotides. Much like the zeros and ones that make up computer code, you have A, C, G, and T making up your nucleotide code that will code for very different proteins, which we'll discuss later. DNA makes up very long molecules. RNA is more often than not single-stranded. It has ribose as a sugar as opposed to deoxyribose. Hence RNA, ribonucleic acid, as opposed to DNA, deoxyribonucleic acid. They have the molecule uracil instead of thymine. Generally speaking, RNA is shorter than DNA, and often RNA is used to code temporary messages because it is less stable. Last, a special use for nucleotides is an ATP, adenine triphosphate. You see here an adenine group with an adenosine nucleotide and your ribose sugar attached to 3 phosphates put in a row, triphosphate. This is relatively unstable to have 3 negatively charged things so close to one another, and thus one of those phosphates wants to leave. To make ATP takes energy, and what you have in ATP is chemical potential energy. That chemical potential energy can be used to do work. Remember potential energy from last lecture. ATP is a way of moving chemical potential energy around a cell and using it for work. And we are going to see that in the metabolism section. So that is it for pH and biomolecules.